In the ongoing global campaign to prevent, treat, and cure HIV and AIDS, there have been countless therapeutic approaches targeting the HIV virus and everything it interacts with on the path to infection. But after decades of relentless infectivity claiming over 25 million lives, what if we could one day simply edit and delete the HIV virus right out of existence? This is ReachMD. I'm Dr. Matt Birnholz. My guest today is leading a novel research effort to leverage gene editing technology in the fight against HIV, and the results so far are exceeding all expectations. He's Dr. Kamel Khalili, professor and chair of the Department of Neuroscience, director of the Center for Neuro-virology, and director of the Comprehensive Neuro-AIDS Center at the Lewis Katz School of Medicine at Temple University. Dr. Khalili, welcome to the program.
Thank you very much.
It's great to have you with us. So before we jump into the research and the technology, I really want to get a sense for where this idea to edit viral genes out of infected cells came from, because while it sounds intuitive and a lot more direct than our suppressive efforts to make HIV a chronic disease, it's also a completely different approach.
Yes, as you mentioned, the current AIDS therapy, which is called antiretroviral therapy, controls virus replication in T lymphocytes of HIV-positive patients, but has failed to eliminate virus from the infected cells or permanently inactivate the viral genome that is integrated in the host T lymphocytes. So, as you know, that once the virus infects human cells, the viral genome, which is RNA molecule, become a DNA, and then DNA become part of the host genome. So at this stage, the virus utilizes the host machinery to replicate their genome, and the outcome is production of HIV in the cells and eventually destruction of the cells. Now, there are small populations of the T lymphocytes retain virus genome, incorporate the virus genome in a latent stage. These cells are called reservoir, usually memory T cells, or the one that retains this basically latent virus genome. So every time that the patients stop taking antiretroviral drugs, the virus rebounds, and then viremia happens, and again go back to the stage of moving toward the AIDS. Now, the way that we can stop this event is inactivating viral genome or excising entire viral DNA from the chromosomes. So we look at the problem as a genetic problem, not infectious disease problem. To this end, we try to find a way to excise integrated copies of the viral DNA from the T cells. So we use a gene editing strategy, which over the last 2 and 3 years received special attention by the biomedical community, so-called CRISPR-Cas9 gene editing. It was about 3 years ago, and we customize, refine this gene editing to specifically identifies and recognizes integrated copies of HIV in the cells, and then apply this technology to the cell culture, human cell culture, in the laboratory. And these cell culture cell models had 1 or 2 or 3 copies of the HIV integrated in the cells. So with our great surprise, we realized that the technology has capacity to eliminate the entire viral genome from the cells, basically converting the infected cells to become completely uninfected cells. So we verify this excision strategy by whole genome sequencing. Basically, we took the eradicated cell and sequenced the entire gene of the treated cells, and we realized that there is no footprint of the HIV coding sequence in the gene after excision. It basically became a complete, if I call it, a sterile cure in the cells. Then we move forward and we ask the question whether or not this excision strategy is harmful for the cell. Does it really introduce any mutations or any gene alteration in the host genome, is it safe? So for that one, we also look at the sequence analysis that we obtain from the cured cells, and compare that one to the untreated cells. And after a series of analysis of the data, we realize that the strategy does not introduce any mutations which could be ascribed to the excision strategy. So it's a safety on the genotoxicity was in place, so there's no genotoxicity. Then we look at the other indices, such as the cell cycle, cell proliferation, cell viabilities, and we realize that it's really safe, at least in the cell culture model, and it does not have any collateral damage to the host genome or cell functions.
And Dr. Khalili, I'll be interested in learning more about the safety element, because that's obviously the heart of the matter for many of our viewing and listening audience. But let me go back to the CRISPR-Cas9 technology that you guys, your team, has leveraged. Most people think of CRISPR-Cas9. It's come on the scene in the last few years. It's gained a lot of traction and interest and appeal within the medical community. But most people think of it in terms of editing so far as being able to address heritable diseases or move in on adding genes where possible, almost like designer genetics. What your team seems to have done is pivot on that concept and think about removing deleterious genes added by infectious disease. So my question to you is, where did that pivot come from? Because there's a pretty remarkable change in sentiment or outlook. And then the second to that is this type of approach limited to targets that insert their DNA into host cells like retroviruses, or are there even broader utilities?
I think that the answer to your question is that, as I mentioned, that the viral genome is integrated in the host gene, and then if we reason that if the approach needs to be taken toward the cure of AIDS, we either have to eliminate all the infected cells, or even better, we eliminate the virus from the infected cells. And the only way that we thought that we can do it precisely without any effect on the host function is this CRISPR-Cas9 system. So we look at the other gene editing approaches, like a zinc finger or talons, and realize that those are very laborious, very expensive, they're very time-consuming. And, we started that, but soon after knowing about the capacity of the CRISPR-Cas9 system, we shift quickly toward this technology, and we were able to, within less than 3 months, get the first set of results showing that the capacity of the system in removing the genes. Now, with respect to the second question, we actually right now using this system for 3-4 other human viruses, and actually one of them which we published several months ago is on a human virus, so-called the JC virus. It's a human neurotropic virus which reactivates in brain and causes a fatal demyelinating disease, or PML. And the virus is replicating in the episomal stage. It does not integrate in the host chromosome. And we developed the technology based on the CRISPR, and we were able to target various regions of the JC viral genome and inactivate the virus in the cells, replication of the virus in the cells. So that tells us that once you have an effective molecule which targets the gene of your interest, either in episomal stage or integrated stage, you can basically target those and edit or inactivate or introduce any alteration in the genes. I think utility of the CRISPR-Cas9 goes way beyond just the HIV and JC. You can apply that one for a number of other infectious disease and HPV, herpes simplex virus, and so forth.
That's fascinating. Well, for those who are just joining us, this is ReachMD. I'm Dr. Matt Birnholz, and I'm joined by Dr. Kamel Khalili from the Lewis Katz School of Medicine at Temple University. we're talking about a whole new potential therapeutic approach in the fight against HIV and AIDS. So, Dr. Khalili, you had talked about your experiments, your research right now being done ex vivo, using cells grown in culture, using T lymphocytes of infected patients. I want to then imagine broadening the scope of gene editing here to millions upon millions of infected cells in the body. And I think the first basic concern that might crop up for those on the bedside of research, on the clinical side, would be how to reach enough of these millions upon millions of cells in the body to fully eradicate the virus. And then from there, how to ensure that the editing goes right in enough cases not to take down the T cells being fixed. I mean, are those even legitimate concerns?
Yes, yes. Actually, the way that we look at this problem is that should gene editing using CRISPR-Cas9 enters into clinic, that's our goal, should be done in the context of the antiretroviral therapy. So ART suppresses virus replication. So basically brings it down to the almost undetectable level in the patients. So, but it does not eliminate the virus from the genome. So what if you find an effective delivery system, that's basically gene therapy, that allow us to deliver CRISPR-Cas9 in vivo throughout the maybe weeks or months, we should be able to reach out to every single cells and the tissues and eliminate the viral genome. One thing we learned was that once you have this CRISPR-Cas9 in the cell, it's going to eliminate the DNA. It has a good capacity to edit the gene. It's a very powerful technology. So I think that with that notion, maybe the technology, the excision molecules will be paired with the antiretroviral for maybe 2 months, 3 months, 4 months. These are some of the estimate that we can get it through the earlier stage of the clinical trials or preclinical studies. And then with notion that once you remove antiretroviral, there won't be any virus to rebound because of the excision has removed the viral DNA from the host genome. So I think that is the strategy that is going to probably work should this technology get into the clinic. And of course, the preclinical studies in the primates is going to tell us far more information. I think that's what I believe that's going to be a way of move forward.
You know, one of the most maddening aspects of HIV is its exceptional adaptability, its ability to mutate. How does CRISPR-Cas9 potentially address that? Would there have to be an ongoing vigilance of that mutational capability, let's say, hypothetically, in each individual patient?
Yes. Actually, if you look at the areas that the virus is mutated, mostly in the area like a GP120, which is coding for the GP120 codings, which is envelope proteins and area. What we did in the initial stage of our studies, we performed bioinformatic studies of all the virus isolates has been sequenced so far. And then based on that, we identified the several areas within the virus coding, non-coding area, which is LTR, which is highly conserved. So we target those areas within the virus with notion that this probably is going to be a universal, basically a target for editing by Cas9. And that's one. So that's the way I think you can probably overcome the problem associated with the mutation. Nevertheless, the CRISPR-Cas9 is a very flexible system. So let's say that if we use our universal CRISPR system in the patients and we realize that you can drop the virus titer by 90%, but still 10% is not responding to your therapy, you can quickly personalize your system. The way that you can do is that you harvest the blood from the patient and then look at the virus, which is derived from that particular patient, quick targeted deep sequencing, look at the gene of the virus, and develop the strategy which targets specifically that population of the virus and then put it back into patients. And this is not very complicated. It's rather with the current technology, with the targeted deep sequencing, and with the creation of the guide RNA in the context of CRISPR, one can achieve the goal within the estimation of the 2 or 3 months. Almost the same thing that we are doing these days in some of the cancer treatment. We are personalizing cancer treatment based on the genetics. So we can basically do the same approach for the virus. Well, except that in this case, you're looking at the viral genome. In the cancer, you're looking at the cancer cellular gene, which is mutated, becoming cancerous.
I am hearing definite parallels there. I'm sure a number of our listeners are as well. So where do you think some of the challenges may lie in utilizing this technology in vivo? I mean, for instance, could the innate immune system be a help or a hindrance, or the lymphatic system, or other aspects that might pose barriers to actually getting the delivery right?
Yes, that is, delivery is something that we need to work on it. And actually, there are a number of gene delivery systems that one can use. We can use a virus-based gene delivery system, such as the adeno-associated virus, AAV, which receives a lot of traction these days. And actually, there's more than 9 clinical trials for different diseases using AAV for delivery gene of their interest. And also nanoparticles is another way you can develop the nanoparticle for delivery of the CRISPR-Cas9 cells. So both approaches we are exploring, and we have a very promising results with the AAV in the small animal model, and we are also utilizing the nanoparticle, which is basically decorated with the ligands, which specifically recognizes the infected cells for delivery of their cargo, which is in this case is CRISPR system. And I think the effective delivery is important. I think that's something is doable, and I think it's going to be probably going to hear a lot of advancement in the gene delivery using those AAV and nanoparticle. And with respect to the question is, do we have to really eliminate every single copy of HIV from patients? What if we just eliminate 70%? And if we eliminate 70%, maybe we can reduce toxicity associated with the presence of the virus in immune cells. And then what happens is you empower immune cells by removing a large number of the virus from the cells. And then when the immune system is empowered, may eliminate the remaining of the infected cells. These are some of the things that we will learn throughout the clinical trials. But there are also good examples. As you know, so far there's only one person got cured from the HIV AIDS, and in this person basically what happened was that through the bone marrow transplantation, and they introduced basically transplanted with the cells which were CCR5 negative. CCR5 is a co-receptor for HIV, and then as of today, there is no evidence for the virus replication in this person. But when they did the bone marrow transplantation, they didn't really remove entire infected cells from every site in the patient. They removed what is the circulating cell was. So that way you're basically empowering your immune system in the patients. So that probably took care of the remaining of the infected cells which were not cleared or substituted with CCR5 negative cells. So I think the strategy of the elimination of the large number of the infected cells by effective and robust delivery system might be sufficient to take us to the cure stage.
And it's an interesting comparison you drew to bone marrow transplants, which compared to this sort of sounds like the brute force tactic or the everything but the kitchen sink out. This sounds far more precisioned, and I'm very, very curious to find out, to your point, whether that sort of immunological herd effect would take place if you remove, say, 70%, and if the delivery's there. Do you think that the delivery vehicle will be the largest milestone to reach in terms of being able to take this to prime time?
If you ask me about that question about 2 years ago, when we had our first publication, I would say yes, probably. But I'm far more optimistic because we have some very promising data in vivo small animal model that shows that actually you can deliver to every single organ ranging from brain to kidney, lung, and lymph node, spleen, and heart, all the tissues in the animal model, and you can actually edit the gene, which in this case was HIV. And I think that we do need to refine the system, but I think we are not that far from that, and we can actually get to the stage of the good and effective delivery system. But again, you never know that what other problem we may face with that. But we have to be basically working on every aspect of it in the laboratory. I think delivery system issue is going to be resolved within the next 2, hopefully 3 years.
We're almost out of time, Dr. Khalili, but just if I were to get your optimistic crystal ball sentiment, what do you think, 5 years, 10 years out from clinical application?
It's hard to say. I think the clinical application, I can't say that, but what we are planning to do, we have right now in a position based on some of the preclinical data that we are getting, maybe in 2 years, 2.5 years, enter into clinical trials, phase 1A clinical trials. We're very optimistic about that, but then we have to see what the outcome of those clinical trials are. But I can tell you that it's very potent, very powerful technology, and for the first time we have been able to eliminate the virus in the laboratory, and it's a promising data, very good data in the animal models. Who knows, maybe in less than 10 years we won't have any problem with AIDS.
It's a great parting comment, a great sentiment to close this particular discussion. And with that, I do want to thank my guest, Dr. Kamel Khalili from the Lewis Katz School of Medicine at Temple University. We've been talking about new applications for gene editing technology in the fight against HIV and AIDS. I'm Dr. Matt Birnholz. Join us at reachmd.com to access this and other research updates. And, of course, tell us what you think. We look forward to hearing from you. Dr. Khalili, thanks again for your time.
My pleasure. Thank you.